going to talk about something absolutely critical in clinical chemistry, HIL interference. And we're going to see why the most important part of getting an accurate lab result often has nothing to do with the fancy machines and everything to do with the sample itself, right from the start.
I mean, think about it. It's the question that every single patient and doctor depends on. You get that report, you see the numbers, but are they right? Can you really trust them? What if the result was wrong before the analysis even began?
And that right there is the heart of the matter. Modern lab equipment is amazing. It's incredibly precise. But here's the thing, it can't fix a bad sample. These pre-analytical errors, as they're called, are a huge and honestly often overlooked challenge in getting healthcare right. So let's meet our three main culprits. We've got hemolysis, Icterus, and Lipemia. And we even have a handy acronym for them, HIL, or HIL. Just look at these tubes. You don't have to be a lab tech to see that something is seriously off here. Their color, their clarity, it's just completely different from a good, clean sample. All right, let's start with the biggest one, the most common offender of the three, hemolysis. This is the one that labs have to deal with day in and day out.
So what exactly is it? Well, hemolysis is when red blood cells literally break apart. They rupture and all of their insides, all those proteins and electrolytes, just spill out into the surrounding serum. And that is what gives the sample that really distinct red or pinkish color you're seeing.
And what's wild is how easily this can happen. We're talking about things as simple as handling a tube too roughly, using a needle that's too small for the blood draw, or even just waiting too long to process the sample. And the impact? It's huge. For example, a hemolyzed sample can make it look like the patient's potassium levels are dangerously high. That could lead a doctor to diagnose a serious condition the patient doesn't even have and start treatments that are totally unnecessary and potentially even harmful. And this is not some rare occurrence, okay? Hemolysis is by a long shot the #1 most common pre-analytical error that labs see. So preventing it really has to be a top priority for anyone handling blood samples. Okay, so we've covered hemolysis. Now let's move on and take a look at the other two troublemakers, Icterus and Lipemia, and see how they can mess with our results. And this visual just shows the difference so clearly. Over on the left, you've got that icteric sample. See how it's a dark yellow, maybe even a little greenish? That's from way too much bilirubin in the blood. And then on the right, that milky, cloudy-looking sample, that's lipemia, and it's caused by a high concentration of fats or lipids. Now, both of these can absolutely throw off the analytical process, but they come from very different places physiologically, and they interfere with tests in their own unique ways. If we look here, we can see that icterus is usually a sign of something like jaundice or liver disease, and it mostly messes up tests that rely on color measurement like creatinine or cholesterol. Lipemia, on the other hand, which you see with high triglycerides, is a whole other kind of problem. That cloudy, milky appearance physically scatters the light that the analyzers use to take their readings. It's like trying to see through fog, and it can throw off everything from glucose to electrolytes.
So we've laid out all the problems, but what do we do about them? Well, the good news is it all comes down to having solid procedures and following good lab practices. Let's walk through that path to accuracy.
This workflow is everything. It all starts at step 1, a proper blood draw. From there, it's as simple as a quick visual inspection. Then you check the automated HIL index that most analyzers give you these days. If there's a problem, you have to document it and flag the results. And finally, if the interference is just too bad, you have to make the call to get a new sample. It's really a 5-step safety check to make sure the results are right.
And that really brings us to the single most important point of all of this. It's a simple idea, but it's the absolute truth in laboratory medicine. It doesn't matter how advanced your technology is, accurate results always start with quality samples, period.
So, to wrap this up, I want to leave you with a question to chew on, especially for those of you out there working in healthcare. After everything we've talked about today, what is one practice, just one small change you can focus on to protect the integrity of the very next sample you handle? Because that right there, that's where accuracy begins.